I'd rather try to give you a different perspective from the point of view of a modeler and sharing my thoughts with you. So just a few, most of you probably know more about this, so I will keep the introduction very short. Also, Dov and Rachel have, and Chris has already made some very important points. So one of the observations we have to consider is that we find antibiotic resistance in people who have never taken antibiotics or for a very long time ago. So this is unexpected from the more expected case where, of course, if you're using antibiotics, we expect to find resistance because we are selecting for it. That, of course, is very clear. It's not just a correlation, it's causation. But it's less clear how you get the antibiotic resistance when you're not using antibiotics. The easy answer is somehow it has to get through the mouth into the gut, but exactly how this happens is a different question. There is an interesting paper showing that actually when you sterilize food, you can reduce the level of resistance. So this is some indication that, at least to some extent, it's based on food. And a recent study has shown that you find resistance genes even in the Yanomami people who have not previously been contacted by humans. Of course, they have been contacted by animals. They've been contacted by bacteria before contact with humans. And what they found in studying the microbiome in these people is that actually the isolates of E. coli were sensitive, but they had the genes. So that's an interesting question of what happened here. One suggestion is that the genes are silenced. So you find antibiotic resistance everywhere. And this is maybe an extreme example. So we have antimicrobial resistance to various degrees, but globally. And the basic idea is that if you're stopping to use the antibiotic, the prevalence of resistance should decline because of the fitness cost of carrying the resistance gene. So this is our hope. The fitness cost of the resistance will bring down resistance if you're stopping the use of antibiotics. This is a very basic idea which is very important. And of course the corollary is that if you use more antibiotics, you will select for more resistance. Bruce Levin has been developing models for a long time to explain this. And this is a prediction of a simple model published more recently by Bruce Levin and others. Assuming a fitness cost of 5%, you can see that the frequency of resistance can decline over time when the treatment frequency is low enough, but this takes years. It would take even longer if the fitness cost would be less than 5%. So it's a very slow process, and it doesn't decrease to zero. Only if time goes to infinity would this go down to zero. So the resistance doesn't actually vanish completely. If you keep treating with antibiotics for some time, you end up with a stable level of resistance gene in the population. So regardless of where you start, you end up with the same kind of steady state of resistance in the population based on treatment frequency and fitness cost and a few other parameters. A recent meta-analysis of many studies published by Brian Bell et al. Is actually from Nottingham, this paper, has looked at many papers, and there is a clear association between the antibiotic consumption and the level of resistance. So clearly the basic idea in this theory is correct. But I will try to explain why this is a little bit too simple. So there are examples where discontinued use of antibiotics has actually worked. So in Finland, for example, they reduced the treatment with erythromycin and the resistance declined. Or in Israel, where they reduced the consumption of ciprofloxacin, the resistance also declined, in line with what we expect from the theory. But there are other examples where it hasn't worked. So for example, in the UK, sulfonamide prescriptions went down a lot, but resistance actually went up. And the explanation here is that the resistance to sulfonamide is carried by plasmids which also have other resistances, which are continue to be selected, so the sulfonamide resistance does not decline at all. Another example in Sweden, where they decreased the use of trimethoprim, there was no decrease in the resistance, a slight increase in fact. Here the explanation is also probably association with other resistance genes, but also that there was no measurable fitness cost. So the idea of having this fitness cost that saves us isn't always the case. So sometimes the fitness cost is very low. So just to summarize these things, in many cases, stopping the use of antibiotics does actually lead to a decline of resistance, but it doesn't always happen, and it never goes to zero. So you have a reservoir of resistance, and when you use the antibiotic again, it will go up again. And this is nicely summarized by this quote: easy to get and hard to lose. Paper from Abigail Sawyers and Carlos Amabile-Cuevas from the 1990s. So a lot of what I'm saying actually was very well known in the 90s. There are lots of these papers from the 1990s which explain all of that already. So there are a couple of things that I want to discuss with you, starting with fitness costs. So fitness costs, the problem with fitness costs is that they depend on a lot of things. It depends on the strain, depends on the medium, temperature, type of resistance, type of antibiotic, a lot of things. So the fitness costs vary a lot. And having a simple model assuming 5% fitness cost doesn't deal with this complexity. I think complexity is a common theme here between the different talks you've heard today. And furthermore, fitness costs evolve. Of course they evolve. If there is a mutation that will reduce the fitness cost, that will be selected for. That's very obvious, and it has happened. There are many studies doing experimental evolution that have shown this. Actually, one interesting study by Dionisio et al. Found even an increase of fitness after evolution of the plasmid giving a fitness advantage to the carrier, and you put the plasmid into the ancestor, even the ancestor had a fitness advantage. So we have problems here if this happens. Also, don't forget that plasmids are selfish. If they carry resistance genes, they also have mechanisms to maintain themselves despite the fitness cost by horizontal transfer. And plasmids may have other effects. For example, some plasmids induce biofilm formation. Now, if biofilm formation has a fitness advantage for the strain, the plasmid will be selected despite a fitness cost because it induces the biofilm. So there are lots of things that make this fitness cost a very complex thing to understand, but it's crucial in order. It's our hope to actually get rid of resistance genes. So one point I want to make is a diversity. So in the gut, we have a very large diversity of organisms. And as you know, fitness costs depend on the strain. So if you have many different strains, this will be important. So if you're thinking of the gut of having on the order of 1,000 different OTUs. Now an OTU is a very large number of strains because there's only 97% similarity in the 16S. There's a lot of other things that can be different if the 16S is the same. And if the 16S isn't the same, there's even more that can be different. So we have to think about this OTU is really not a useful way to categorize our diversity in the gut. We really have to think about the strains, because strains are real. Strains can be selected. Strains have an effect. They infect or carry resistance genes. OTUs are nothing real. Also important to remember is that we have an enormous number of microbes in the gut. So given these considerations, let's assume we have something like 100 strains per OTU, so we have 100,000 strains. There are many question marks because I don't know, okay? But it's a large number, I'm sure. And if you're thinking of a broad host range plasmid that maybe can maintain itself in 1% of all these, that makes 1,000 strains. And if we have a probability of this plasmid being beneficial for the carrier of 1,000, we end up with a plasmid that can reside in 1,000 strains and increase fitness of one strain. And this one strain, where the fitness is higher, despite carrying the resistance genes, will be a reservoir that we can't get rid of just by discontinuing the use of antibiotic. So what we have to consider is the variation of costs and diversity of microbes, because this means we will have reservoirs of resistance in the gut and, of course, in the environment, as Rachel told you about previously. These are the reservoirs we have to worry about. And it also means that in our research, we really have to consider the diversity of strains. I think we need to move away from just working with a single strain and a single gene and a single antibiotic in the laboratory. That doesn't cope with the complexity that we have to deal with to really make our approaches work in the real world, which is the topic of theme 3, as you all know, of course. So, another effect of the high diversity is that it affects the spread of the plasmids carrying resistance genes. Again, a paper by Dionisio, so you see I'm a fan of his work, showing that plasmids can actually spread very quickly in a diverse community like the gut. So they studied the R1 plasmid, and the transfer rates of the R1 plasmid vary widely between different hosts. So some donors have a very high transfer rates, and others, because of fertility inhibition, carrying already another F-type plasmid, for example, have a much lower rate. So it's these donors of the plasmid that have the very high transfer rate that lead the transfer in the environment, in this case the gut. So this will speed up the transfer a millionfold compared to strains to donors which have a lower rate of transfer. So what that means is that we have to consider the diversity of transfer rates. An average transfer rate in a model makes little sense because the spread of the resistance is really driven by the extremely high transfer rate. The average would be useless, okay? So we really need to think about the complexity and the distribution of transfer rates, distributions of resistance, and so on. Another point about the gut I already mentioned, we have a huge number of cells in the gut, so that even if the prevalence of resistance is very low in the relative sense, let's say a millionth, that's still an awful lot of bacteria that are resistant. In fact, so many that there's no hope that there will be a bottleneck leading to random extinction. You know, with 10 to the 7 cells, you don't get random extinction. Okay. Another issue we need to think about is heterogeneity, covering all scales. So starting with the small scale of diffusion gradients in the biofilm, for example, or the gut mucosa. And then different concentrations, different sites of the body. Or, of course, variation in time is very important because we don't continuously consume antibiotics at the same concentration. Different concentration, different people, animals, aquaculture, and so on, in different countries. So there are lots of different concentrations and different combinations and different time courses. And all of that leads to different selective compartments. So this is a term coined by Fernando Baquero, Maria Negri, that there are different selective compartments in the body, but this generalizes to the environment, obviously, where you have selection for low levels of resistance in some parts of the body and selection for higher levels of resistance in others. And all of this isn't considered in the simple models that have been used so far. Then you have the hitchhiking, where if a gene is coupled to another gene which is positively selected, like being on the same plasmid, you will select for the gene that's hitchhiking despite it causing a fitness cost. So for example, if a resistance gene can hitchhike with other resistance genes, or in fact any other gene. An example that I found very intriguing is from Ann Summers' work showing that selecting for mercury resistance by having mercury in your dental fillings is actually increasing the resistance for other antibiotics. And maybe one probably more cost-effective than developing a new antibiotic would be to get rid of the mercury fillings of everyone. So we have to really think very broadly about all the factors that can contribute to resistance and its persistence. Being a fan of biofilms, I wanted to mention biofilms a little bit at least. In biofilms, you have tolerance against antibiotics at high concentrations, or most antibiotics at least, despite or even if the cells are not resistant at all. So this is just tolerance. But it gives an opportunity for selecting resistance, and you also have concentration gradients in the biofilm, so speeding up the evolution of resistance. So I think the question, considering all of what I've said, I think just using antibiotics more prudently will not mean that resistance will go away. I don't think there's any hope in that. There will always be some level of resistance remaining due to the diversity and heterogeneity and variation that I've explained. So what can we do? Lots of things actually. I don't have much time, so just a very quick run through this. We can try to use more narrow spectrum antibiotics. We have to think about the dose. Now, intuitively, you would think that a higher dose is the best way to stop the evolution of resistance. And also, you would think that using a combination of two drugs would be better able to stop the evolution of resistance. But recent work by Robert Beardmore's group in Exeter has shown that things are a bit more complicated. He has actually shown in a paper called Smiled-Frown Transition that having a high dose will, in the short term, over one day, lead to less resistance, but over several days will actually increase the level of resistance. So what is a short-term benefit of a higher dose actually ends up as a long-term problem. So just one example to point out that things are not so easy, and just thinking that a higher dose is the best way to do this is not necessarily correct. Of course, we need new drugs, but more importantly, we also need to think more about alternatives to antibiotics, like antivirulence drugs, blocking chrome sensing or adhesion or other things, using phages or the predatory Bacterium delevibrio, which I'm highlighting here because this is also research done in Nottingham by Liz Sockett's group. And this gives us chances to use other strategies or combine with other strategies. We also need to think of directly attacking the resistance genes, maybe with CRISPR or other systems, and preventing the transfer of the resistance genes. So thinking of drugs that inhibit plasmid transfer, there are some fatty acids that have this effect, that they might be considered to be very helpful. And this is a very simple idea, but actually I think it's very important. We've got to think about preventing infections in the first place, because fewer infections means fewer treatment with antibiotics. So this is an early thing you can do, is to prevent the infection in the first place. So we really should do more about this. We should do more vaccination to prevent infections, hygiene, and this is also not just about us, it's also about animals and farms and pets. So we need to prevent infections, and better diagnostics is also helpful if we can work out whether it's a bacterial or viral infection, making sure that we don't prescribe antibiotics if it's a viral infection. And also a point I want to make here is that I think we need a different kind of research than just a single strain, a single resistance gene studying in the lab. We need to consider the diversity of strains and plasmids and resistant genes and the fluctuating concentrations and the evolution, because evolution is extremely powerful and we have no chance to beat evolution. We have to find ways where we can exploit evolution rather than try to beat it, because that's not possible. So we have to really think about the real complexity of the system. And I think we need to use quantitative models to optimize a treatment, not at the level of the individual patient only, but at the level of the population, in fact, the global level. Because it's like climate change is a global problem, and it's not what happens in China. We had a question about China. It does matter for us very much. So I think the complexity we have is too high to deal with this without the help of models. And the simplistic models we have used so far are very useful in terms of understanding and insight, but they are not good enough to really make predictions or use them for management, because you can't use a model for management if you're neglecting a lot of things that we know are important to the outcome of your strategy. So we need models that consider all of these things. So at the end, I want to use a few slides only to give you some idea of what I think I can contribute in this community effort. And basically what I think I'm good at is making models more complicated. And one thing I've studied is the dynamics of plasmid transfer, work done by Brian Murky and Sonia Martins. And I've done a lot of population dynamics modeling currently done by Robert Clegg, but many other people previously. So we have software called Idanomics, which is free for you to download and use, which allows you to model populations of bacteria interacting with each other. And we are working on eGut, which is based on Idanomics, which will, when ready, be freely available for you to study population dynamics, plasmid transfer, and things like this in the gut environment. This is work funded by the NC3Rs. So, I don't know whether you can see this is a confocal image of a biofilm in the flow cell. You can see that the green cells here are the transconjugants. They are only around the surface of the biofilm, so they cannot invade the biofilm itself. And we've used a model to try to explain this observation. Basically, if you assume a growth rate dependence of conjugation, if you have no growth rate dependence, as in this example, the plasmid can spread throughout the entire biofilm. But if you assume a growth rate dependence, so higher transfer rate at higher growth rate, the plasmid is not able to penetrate the entire biofilm. If the biofilm is older and it doesn't grow so fast, you don't get any penetration if the growth rate dependence is too strong. So that doesn't mean that this is the reason why the plasmid is only found in the surface of the biofilm, but it is a potential reason. There may be other reasons. We can't exclude that. Another thing we have done is studying the dynamics of plasmids, competing narrow host range and broad host range plasmids in chemostats and biofilms. I don't have time to go through the details here, just so you can see that it's kind of interesting things happening in these models. And we use the same for studying transfer of plasmids in biofilms. That I do believe is relevant to what happens in the gut environment. A broad host range plasmid clearly has an advantage in spreading through the entire biofilm, the narrow host range plasmid being limited to be carried only in one of the two hosts. And that is a disadvantage for the narrow host range plasmid. And so the broad host range plasmid has a higher fitness despite the higher fitness cost it has. So what we're currently doing is developing eGut, where we are basically having simulations of a small region in the gut, one for the mucosa and one for the lumen. And from these, simulating these small regions in very much detail, you can then scale up to the whole compartment of the gut, and then you can have a chain of compartments to do the whole gut, and then you can have other compartments in the body to be able to model the interaction between the gut and the other compartments of the body. Okay, so that is what we're currently working on. And I'm proud to say that in Birmingham, we are part of the SGM Small World Initiative, the undergraduate program. Anne-Marie Krachler, who is with me in the audience, and I will be leading this in Birmingham. And of course, you all know that we are very active with antibiotic action in Birmingham. And thank you very much for your for listening.